Hello, so my name is Noah Sadlami. I graduated year 12 in 2023, so depending on what time you're watching this, but for me that was a few weeks ago. I was rank 1 throughout the year for biology and the ducks of my subject. So my advice for VC biology is that whilst it is a subject that shouldn't be taken as sort of a rote learning only subject, memorization is very crucial, especially for VC biology. So how I like to think about it is that 60% is memorization and 40% is understanding. So you might think it's the other way around, but in my experience, that's how I saw it. So there's actually been students in my class when I was studying, and we had the same level of understanding. So we actually understood the concepts the same way, but the reason why I scored much higher than them was because I took memorization in a sense that it was more important than they saw it. So in a question, for example, in gene expression, like provide an overview of the steps. So for them, they'd go, oh, the DNA is copied, and then a cap and a tail is added, and parts of the copy are removed, and then that leaves the nucleus, and that goes to a site, and then that is correct. The overview of that is actually correct, but the keywords what they're missing. So they're missing, for example, the keyword of the DNA. The DNA template strand is copied by RNA polymerase. This undergoes processing, and a methyl G cap is added, and a poly A tail is added, and introns are removed. So I've just used about 5-6 keywords that I memorized, and that would get me full marks. Whereas for them, they get 0. Even though the understanding is the same, they had no memorization, and that really hurt them. So what I'm going to do is I'm going to provide you with that 40% to understand, and then I'm going to enclose Anki flashcards which I've made. They're not just definitions, they're definitions, photos, fill in the gaps, and these will help in the memorization aspect. So I'm giving you that 40%, and then the rest is for you to finish that 60% off and constantly revise. And it shouldn't take you more than 15 minutes to do these flashcards. And by the end of the year, you'll see that everything's been memorized already. It doesn't seem like a big chunk of, oh, what's that keyword? It's all been done, if you are consistent with these flashcards that I am going to upload. Now, what we're covering in this topic is gene expression. So the specific study design dot point that we're covering now is the steps in gene expression, including transcription, RNA processing in eukaryotic cells, and translation by ribosomes. Now, RNA processing in eukaryotic cells, it doesn't say just RNA processing, and that's because prokaryotic cells do not undergo RNA processing. This is because prokaryotic cells do not contain introns. So I'll explain later on what that is, but prokaryotic cells do not contain introns, and these introns need to be removed during RNA processing. So if prokaryotic cells do not have them, they do not need to remove them. So that's why they do not undergo RNA processing. But for now, what I want you to remember is that prokaryotic cells do not undergo RNA processing. So prokaryotic cells do not undergo RNA processing. And then the rest of that study design dot point is and translation by ribosomes. So translation is done at ribosomes. So ribosomes are the site of translation. That's basically what that study design dot point is clarifying. So we'll delve deeper into that. Before we actually begin and I sort of pinpoint what gene expression is, for those of you that haven't done Unit 1 and 2 biology, this will be new to you. For those of you that have, this will sort of be a quick refresher. So DNA, this is just brief definitions to explain what sort of keywords we're going to be looking at in this topic. So DNA. DNA is a double-stranded molecule made up of nucleotides. So its monomers are nucleotides. And this carries the instructions, so the DNA carries the instructions for organism or cell to survive. For cell/organism survival. So I'm not sure if you guys have watched Survivor, but imagine that you're chucked on an island and you've got a manual for how to survive. The DNA is basically the manual of how organisms survive. It contains all the codes that produce things called proteins, and these are basically what allow us and other organisms to survive. So the DNA carries instructions for cells/organism survival. Now, a gene is just a section of DNA. So it's a section of DNA that carries the code for a protein or whatever. So it's just a section of DNA. That's a gene. Section of DNA. Now, we're going to just briefly define what RNA is. RNA is ribonucleic acid. So the difference between these is that, so DNA is deoxyribonucleic acid and RNA is ribonucleic acid. So the difference is in the oxygen. So ribonucleic, can't pronounce it, but RNA contains oxygen and DNA does not contain oxygen, so deoxyribose, whereas ribose. So that's the difference. And then there's 3 types of RNA that we're going to be looking at. So there's messenger mRNA. It's commonly referred to as mRNA. And basically what that is, it's an RNA molecule that's produced following transcription. And what that does is it carries the genetic information from the nucleus to the ribosome. So just as the study design dot point clarified, translation occurs at the ribosome. Translation occurs at the ribosome. So, but the information is located in the nucleus. So a messenger mRNA basically it takes that information from the nucleus and delivers it to the ribosome for translation to occur. So I'm just going to give a brief definition. So produced following transcription. Yep. And so that's basically the first thing. Produced following transcription, and it carries the genetic information from the nucleus to the ribosomes. So that's pretty easy to remember. It's just it's a messenger. It acts as a messenger from the nucleus to the ribosomes. Now, tRNA or transfer RNA. So once we're actually at the ribosomes and we're undergoing translation, we need to actually make that mRNA strand produce for us what we want as our final product, which is a protein. So I'm just going to go quickly. I'm going to quickly jump here. So the monomers of proteins are amino acids. So amino acids are monomers of proteins. So that's what proteins are made up of. The subunits, the blocks, are amino acids. So when we look at transfer RNA basically reads the mRNA strand. So reads the mRNA strand and it delivers amino acids specific to the codons on the mRNA strand. So I've introduced a new word, and that's codons. Codons are basically a sequence of 3 nucleotides on the mRNA. So 3 nucleotides on mRNA make up a codon. So the transfer RNA or tRNA that reads the sequence of these 3, that codon, and it delivers a complementary amino acid. So an amino acid that is complementary to that sequence. So it's not just bringing random amino acids and joining them together. Now, rRNA or ribosomal RNA, this you don't really have a lot to memorize for this. It's just a structural component of ribosomes. That's all you have to know for it. You never really questions on rRNA. At most, you find that they ask you what is mRNA, tRNA, and RNA, but you'll never be sort of questioned directly on RNA. Now, we've clarified that amino acids are the monomers of proteins. So yeah, if you add, let's go A plus These signify amino acids. So plus A. What you'll get is a polypeptide. And that basically is a protein. So later on, after the changes to the polypeptide chain, it will become, will go into its structural form and be a protein. So that's basically the overview of that. Now that we've sort of clarified those things, I don't expect you to be experts on this. This was just to sort of open up your head so you can sort of understand what's going on when I begin to explain it. Now, before we begin, I'd like to say that if you have any questions while I'm teaching, while I'm speaking, write them down, note them down. Don't just leave them in your head so that you forget. These questions are very important, so it's better that you ask me these questions and clarify them than not ask at all, and then come SAC or exam time, you see that question and you're just like, oh, that's what I didn't know. So yeah, if you have any questions, even while I'm speaking, obviously I don't expect myself to answer questions whilst I'm explaining a concept, but write them down. Make sure that you write them down so that we can tackle them at a respectable time. So, how are proteins made? That's basically the first thing that we're looking at. So like I said, DNA is a set of instructions that a cell requires to function and survive. So that allows for the production of proteins, which are responsible for essentially all the functional capabilities of a cell. So basically, without them, you're not going to survive. Without them, an organism won't survive. So there are a number of key steps that are required to follow these instructions, known as gene expression. So basically what we're looking at is gene expression, the expression of genes. So the expression of that part of DNA that codes for proteins. That's what we're looking at. Now this can be broken into 3 steps. So the first one is transcription. RNA processing is number 2. Oops, for the and translation. There we go. So, transcription and RNA processing, both of these occur in the nucleus. And then translation occurs, as the study design.point clarified, in the ribosome. Now, what actually occurs during transcription? During transcription, actually before I even explain that, I'm going to redefine DNA. The DNA molecule. So DNA is double-stranded. So that's strand 1, strand 2. Sorry, I'm not the best at drawing. One of the strands is known as a template strand, and the other one is known as a coding strand. Now, in transcription, a specific molecule known as RNA polymerase, so RNA polymerase, that binds to the template strand. It doesn't bind to the coding strand, it binds to the template strand. So, let's say that's around here. So it binds to the template strand. Specifically, it binds to the promoter region. I'll explain that later on, but it binds to the template strand. It reads it from a 3' to 5' direction, and it produces a complementary strand known as pre-mRNA. Here they've just written RNA, but for example, if you're annotating it on an exam or something, you want to say pre-mRNA. You want to be specific. That's where the memorization comes in, which I'm going to include in the flashcards. Pre-mRNA. So the pre-mRNA cannot actually leave the nucleus and go to the ribosome because it's not mRNA yet. Only mRNA can do that. So it needs to undergo processing. So RNA processing. So this is all in the nucleus. So we've finished transcription. That's been done. And now it has to undergo RNA processing. So in RNA processing, first of all, these things are gone. These little green bits. And also a cap is added and all these A's are added. So that cap is known as a methyl G cap. And the tail, those A's, that's known as a poly A tail. So the 3 things that happened is that those green bits, which I'll explain what they are in a sec, those green bits are removed. methyl GCAP is added, and a poly A tail is added. So what are those green bits? Those green bits are introns. So introns are non-coding regions of DNA, and exons, the red bits, are coding regions of DNA. Now, an easy way to remember which ones are used to produce the final protein or not is that introns remain in the nucleus, so they do not leave to go on to produce the final protein, and exons exit the nucleus. So they exit the nucleus because they go on to contribute to the final protein. In an exam, you might be asked, what's the function of introns? This is later on when we learn about mutations and everything, but just to, in case some of you guys are curious, why does a cell or an organism, eukaryotic cells, have introns? What's the function if they're not coding? It's that in case of a mutation, the likelihood of a random mutation affecting a coding region is decreased. So if a random mutation does occur, the likelihood of it affecting coding regions is decreased because of all the non-coding regions. It will be more likely to hit the non-coding region than the coding region. It doesn't make it impossible, but it just decreases the likelihood. So it's sort of a safety mechanism by eukaryotic organisms. Now, when you're asked about RNA processing, those are the three things you have to remember. So removal of introns, splicing of exons together, addition, so those can be one sort of, and then addition of a methyl GCAP and the addition of a poly A tail. Now, you don't actually have to know exactly what a methyl GCAP is or what a poly A tail is, but what you do have to know is why they're important. So you don't have to know what they are, you have to know why they're important. And the idea of them is that it provides stability for the mRNA to bind to the ribosome. So it allows it to sort of bind and be stable on the ribosome. So that's basically the function of the cap, the methyl GCAP. You have to say methyl GCAP, and the poly A tail. Now we have an mRNA molecule. Now that is exported from the nucleus, so it leaves the nucleus, and it goes to the ribosome. So this is now translation. Sorry, someone's incorrectly labeled that. It's translation. We'll sometimes got a couple typos in our content, sorry about that. But yeah, so now translation is occurring, and that occurs at the ribosome. Don't forget that, translation at the ribosome. Now, during translation, tRNA, so these, I'm going to use these as codons. These are the sequence of 3. So 3 nucleotides. So that's codon, 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 codon. Now tRNA is going to come and read these codons, and it's going to, these are free-floating amino acids. Free-floating amino acids. I'm just going to label that as amino acids. So the tRNA is going to read these codons, and it's going to travel and bring a corresponding amino acid. Corresponding, not random amino acids, corresponding. So yeah, so it's going to deliver these. So tRNA is going to come, pick one up, and then deliver it here. And then one by one, they're going to align next to each other and join. And once they join, they form a polypeptide, what we know as a polypeptide. So that's the amino acids joining together produces a polypeptide. And that's what we're talking about. The monomers or the subunits of proteins are amino acids. So when you join them all together, you get a polypeptide, which in turn produces a protein. And then, now, how could this process be different in prokaryotes? So in RNA processing, we said that the introns are removed. Now, in prokaryotic cells, they do not contain introns. They only contain exons. So RNA processing does not occur. So in prokaryotes, they do not contain introns, and therefore RNA processing does not occur. And then why would RNA be a useful intermediate to have between DNA and proteins? So this all works in aiding in the regulation of the amount of proteins produced. So for organisms, they do not want to overuse their amount of energy. So they don't want to produce a lot of proteins because that requires energy. and they want to conserve energy. So conserving energy is vital for organism survival. So having RNA as an intermediate, it sort of acts as a regulatory process. It can act as a regulatory process along with other gene regulation things. But yeah, it can act as that as well. This is pretty broad. You can tackle it in multiple ways, but that's the way that I've chosen to tackle it. Now, moving on, we're going to section B. We're going to look more deeply into transcription, which is step 1 of gene expression. So, transcription is a process by which an mRNA copy of DNA is made in the nucleus of the cell. That is correct. A complex multi-step process, it is initiated by a key enzyme. So, that key enzyme, as I've mentioned prior, is RNA polymerase. RNA polymerase. Now, what do we know about enzymes? This is, I'd say there's 3 important things to know about enzymes. Number 1 is that they are organic molecules, or organic catalysts, actually, you could say, which is even more specific. And what they do is that enzymes, like catalysts, they don't produce new reactions. So it's not like you're producing a reaction that wouldn't occur, but you're speeding up a reaction that would occur without the enzyme, but you're just speeding it up. So it speeds up the rate of reaction. And how does it do that? So VCA looks for that extra bit. So you can say, it speeds up the rate of reaction. You probably, if it's out of 1 mark, you probably won't get the mark. You have to say by decreasing the activation energy required. The third thing that I'd say is important about knowing about enzymes is that they remain unchanged following a reaction that they aided. So remains unchanged. So yep, it remains unchanged. Now, VCA does not assess transcription in these steps, but it might make it easier for you guys to tackle it. So we can break it down into 3 steps. Initiation. Second one is elongation. And third one is termination. To begin the process, transcription factors assist in the binding of RNA polymerase to the promoter region. I've mentioned that before, but now I'll more properly explain it. So if in an exam or SAC you're asked, what is a promoter region? All that you have to say is that the promoter region is the site at which RNA polymerase binds. That's it. That'll get you the mark. So that's the promoter region. So RNA polymerase binds to the promoter region. That kickstarts the transcription, but it has to bind to the promoter region. Now, this initiates the unwinding of DNA, achieved by breaking the hydrogen bonds. For those of you who do chemistry, we know that what holds DNA together is hydrogen bonds between the 2 strands, allowing the formation of the mRNA strand to occur. So, this diagram, since we haven't really explained what the termination site is in 3', 5', I'm not going to delve into right now too deeply, but what I am going to say is the most important thing about this diagram to take on is this. RNA polymerase attaches to the promoter and separates the DNA strands. From this diagram, don't worry about the rest of the stuff. We're going to look at that deeper. What you should be looking at is RNA polymerase attaches to the promoter and separates the DNA strands. If you're asked how it does that, hydrogen bonds, by breaking the hydrogen bonds. Now, elongation. What happens in elongation? So RNA polymerase then moves along the template strand. Like I said, the template strand is what is read by RNA polymerase, and a complementary strand is produced from the template strand. So, and then free-floating nucleotides will come and base pair with the exposed template strand. Now, RNA polymerase will catalyze the condensation polymerization. whenever you see this term condensation polymerization, it refers to the byproduct of water being produced. So condensation polymerization. To join the sugar phosphate backbone. So the sugar phosphate backbone is sort of what holds the DNA together. So that's basically the overview of that. We're going to go deeper into condensation, polymerization, and all these terms. But as of right now, since we're just sort of providing an overview of gene expression, that's all that you have to know. That condensation, polymerization occurs, and it rejoins remakes the sugar phosphate backbone. Now, it forms a pre-mRNA strand, not mRNA, pre-mRNA. Now, as the RNA polymerase moves along, it rezips the section of DNA that has already been transcribed. That's great. Termination, the elongation process continues until a termination sequence is reached at the end of the gene. So, the termination sequence basically it tells the RNA polymerase when to stop expressing that gene. Because otherwise, how will it know? RNA polymerase needs a signal, and that termination sequence acts as a signal to halt it. The DNA will rezip and rewind, returning it back to normal. So the DNA remains unchanged. Great. Okay, what is the relationship between the new mRNA strand and the DNA? Okay, I'm going to tackle this from the very beginning. So, from the DNA, RNA polymerase reads the template strand. So I'm just going to say DNA template strand. Template strand. I'm going to just draw this. And then I'm just going to say it produces pre-mRNA, about the same size. So that's pre-mRNA. This pre-mRNA is identical to the coding strand, but it is complementary to the template strand. So the template strand and this strand do not contain the same nucleotides. Do not. Don't make the mistake of saying that they're the same. They do not. They are complementary. The term is complementary. Not the same, complementary. Now, that pre-mRNA, obviously, introns are removed, addition of caps, blah, blah. So cap, and then A. As you can see, I've scaled the mRNA strand to be much smaller because obviously introns are removed. So the question now is, what is the relationship between the new mRNA, so this is the new mRNA, not this, that's pre-mRNA, and the DNA? A simple way could be that it contains nucleotide sequences found in the coding strand. And that's basically a way that you can answer that question. But obviously, if you're struggling with a question, so for example, if you've just seen this question, you might not know how to tackle it straight face on. So try to draw it out. What's actually happening to the DNA strand, and how do we get from DNA to new mRNA or the mRNA, so not the pre-mRNA? What direction is transcription occurring, and why is this the case? So transcription occurs from the 3' to the 5'. And the reason for that is because the promoter region is located in the 5'. And this is the template strand. And the termination sequence is located at the 5' end. So remember that the promoter region is the site at which RNA polymerase binds. So RNA polymerase, let's go RNA polymerase. And the termination sequence is what stops the RNA polymerase from continuing to transcribe that gene. So now it goes past exam reports can be an amazing resource to determine what the correlating ones in their answers. They are the perfect answers in their eyes. So yeah, an important note is that obviously the way that I'm teaching is to improve your understanding. So in an exam, you won't write initiation, elongation, termination. That would, 1, consume too much time, and 2, that's not what VIC is really looking for. They're looking for basically the ideal answer, which in the flashcards that I'm going to enclose, it includes the ideal answer. So it's not going to include like a big chunk of information that just makes you understand things better. Like obviously in an exam, you're not going to write, oh, the DNA is the recipe and the proteins is the cake. That just helps you understand things better. but in an exam there's ideal responses, all of which I'll enclose and support you guys with. Now, for example, in this question, explain what RNA polymerase is and its function. So you can pause this video and give it a go by yourself, and I'm going to start to explain how I would tackle this question. First things first, which a lot of people don't do, is look at how many marks it's asking for. So if this was asking for 3 marks, I would include the direction of movement, when it stops at the termination sequence, all these things, but it's asking only, it's only providing 2 marks. So I'd look at where can I get these 2 marks from? So explain what RNA polymerase is. It's telling you what to do. Explain what RNA polymerase is. That's 1 mark. Second mark, its function. So let's tackle this. So RNA polymerase is an enzyme. That's basically what it is. And what's it doing? So first of all, that binds to the promoter region. Remember, that's where RNA polymerase binds. Region. And transcribes the DNA template strand. So that's sort of half of what it does. So it does transcribe the DNA template strand, but what does that do? That in turn produces a pre-mRNA molecule. There we go. That would get you 2 marks. So you've explained what RNA polymerase is. It's an enzyme that binds to the promoter region, and what it does is 1, transcribes the DNA template strand. 2, that in turn produces a pre-mRNA molecule. Not an mRNA molecule, pre-mRNA molecule. And that is all that we have for today. If you have any questions, like I said, hopefully you've been writing them down. Send them through. We've got, I think, about another 4 minutes of this lesson, but I don't mind staying a bit extra. So yeah, ask me what these questions are, and we'll tackle them. If you want to ask me more privately, or you want to email me later on, I'll get back to you as soon as possible. And yeah, please do these flashcards. They shouldn't take you more than 15 minutes each time. If each lesson that we have, you do those 15 minutes of flashcards, by the end of the year, you'll find that it's so easy, and the revision that you have to do has already been done. You won't have to re-revise or anything. So please take these seriously, and yeah, hopefully you enjoyed this lesson, and we'll see you in the next one.